So welcome everybody to this session. Thanks also for the organizers to organize this wonderful MPS conference. We almost doubled, I would say, in the participants this year compared to last year. It's quite amazing. So I would like to give you some insights into some models we recently applied and developed internally for CNS. But before I show you the models, I would like to start with actually the why, right? So why do we as Roche think we need to focus on complex in vitro models and advance those? And there is intrinsic factors, but there is also extrinsic factors. So the intrinsic ones are clearly a shift in the portfolio from the past being a small molecule-driven business towards today, which is mainly, or not mainly, but the majority of a lot of molecules are large molecules and other modalities such as RNA therapeutics. And the speciality of these newer modalities, right, they have other specifics and they do not necessarily, and they are partially so specific to the human target that they do not cross-react anymore with any animal models. So we have to come up with some model systems, or we had to come up with some model systems which recapitulate the human situation where we can do sufficient safety testing. And then also testing these novel modalities or new molecules in an in vivo situation would also be unethical. But as said, there is also extrinsic factors, and I think most of you are aware of this one. So we have a lot of challenges with monkeys, which is unfortunately one of our main animal species for safety testing in industry. And there were supply constraints also triggered by COVID partially. So today we have to plan some of the monkey studies 1 to 2 years in advance and reserve slots at the CROs. But we also, there is a strong push from society, obviously, to move away from these monkeys. Air France, for example, stopped research transport of monkeys. And then also cynomolgus monkeys has been labeled as an endangered species. So just to nominate a few, I mean, there is a clear need that we have to move away and to look into alternatives, which we all do, and we saw over the past days beautiful examples how nicely we advance already in the field. But before we can entirely replace that, we also need to consider what are we going to replace for safety testing. And this is actually this, as an example. This is a 4-week small molecule toxicity study in a rodent or non-rodent. So I don't go into all the details, but taken together, 4 weeks testing in an animal, we measure more than 130 parameters, being safety, being PK/PD, being ADME, and others. So, I mean, if we really want ultimately replace entire animal studies, this is something we have to come up, and this is an opportunity, but also a challenge for the entire field. And I guess this is one of the reasons why we are here today. So now, or today and in the past, we mainly used in vitro models in these two clusters, we could say. And this is a nice graph from a publication of Adrian Roth and Thomas Singer from 2014, and I think this is still true for today. So we have predictive in vitro models where we would like to ultimately and directly predict from the in vitro situation towards the clinical outcome in the patients. But we also use them as mechanistic in vitro models. So we would like to understand not only mechanisms of toxicity, to understand it and come up with better molecules, maybe better targets to avoid those, but also understand if our animal findings in the safety study are truly human relevant. And I will show you 2 of those examples in the next few minutes. So first is a predictive in vitro assay. We established in-house about front of the eye. So thinking about the eye, most of you, also in the context of CNS, you would think about the retina. But obviously there is multiple compartments of the eye, and one of them is the cornea. And having a corneal toxicity could also lead to severe impairment of the visual acuity of patients. Therefore, it's important to test for this liability. Luckily, we had the opportunity that there is already a model existing for corneal toxicity testing, an in vitro assay, which is since 2020 an OECD accepted test method for eye irritation. It's a reconstructed human cornea-like epithelial model, and it's used for chemical testing for eye irritation. So we started looking into this model, and we asked the question, is it also relevant for pharmaceutical testing? Because the chemical testing is very acute, whereas pharmaceutical testing is mostly repeat dosing and a longer exposure times usually. So before treating the model, we characterized it, and our histopathology analysis revealed that the model nicely recapitulates the human cornea epithelium with all the multi-layers, with all the sufficient cell types like basal cells, wing cells, and superficial cells. And it was a bit thicker, and it contained a few more apoptotic cells, but nevertheless, histomorphologically, it resembled the human cornea to a very high degree. And bulk sequencing showed that, yeah, it was a little bit more towards the conjunctival tissue, but still we consider the model is a very relevant model resembling the human cornea for in vitro testing. So we started with the standard protocol for eye irritation testing, and we applied some of our compounds A to E or F, and we could reveal after an acute dosing of 30 minutes with a watch-out that only one compound was rendered positive, meaning eye irritant. However, then we applied a more physiological or more in vivo-like paradigm, and then we could show that actually 3 of our compounds turned out positive. So, which showed us that indeed repeated dosing, and we call this the cornea repeat dose assay, which you may have seen at a nice poster of Karen yesterday, is more relevant because it resembles more the in vivo-like setting, and it also increases the sensitivity. So we could show that one of our compounds, which had severe findings in an in vivo study for 14 days, is also rendered positive in the assay in a dose-dependent manner. But it's not only testing tool compounds or in vivo compounds in the in vitro models, but also we need to consider about assays and readouts which are translatable between the in vivo situation and the in vitro situation. For that, we applied a cornea fluorescence staining, which is a kind of a standard method to diagnose patients in clinic, but also animals for corneal impairment. And we transferred this assay to the in vitro model. And what you can see here again with our compound E and with another positive control compound, that indeed we can recapitulate with the assay eye irritation in our model. And with that, we have established an assay which is not only applicable in vivo, but we can also apply the same methodology in vitro. So what is next? If you think about front of the eye safety assessment, it's not only about the cornea epithelium, but it's more than that. And we learned during in vivo studies that also the goblet cells is one source or one target for toxicity. So we made use of a surrogate model, which is an air-liquid interface model, which you can see has nicely developed goblet cells, which you cannot keep isolated in culture, unfortunately. And we applied again a test compound which has previously shown to be toxic on the goblet cells, and you can see that it nicely, in a dose-dependent fashion, kind of kills the goblet cell in the model, which was quite promising. So we are currently looking further into this goblet cell model as and the Ali model as a surrogate model for goblet cell toxicity, and will further test it with more tool compounds in our setting. So now we switch from a predictive in vitro assay more to a mechanistic in vitro assay, which is in that case the brain. And we don't need to go through the details. I guess you have seen many nice examples over the past days here about nice brain models, which all need to have a complex cytoarchitecture, and they need multiple cells to become relevant for brain physiology. In our case, to show that these models are indeed relevant for what we want to assess is that most of the data we had in this platform project was a non-human primate data. which render the question if we would not need also a non-human primate model. But before we came there, we established a protocol in-house which is derived or based on iPSC cells, which turn into brain spheroids after 8 weeks of growth. And we could also integrate microglial cells, which you see here on the movie, which nicely home into this spheroid over the 8 weeks in culture. We also established an assay based on high content imaging and caspase 3/7 activation to show, based on this imaging, that we can measure even nowadays over a time frame cytotoxicity in this model, and we could show that this is also, it captures classical neurotoxicants. The human assay or the human model nicely shows oligodendrocytes, astrocytes, neurons, and even microglia in a homeostatic state of over multiple days in vivo, and the maturation markers nicely increase with time. Also, the microglia responds to a toxin-induced demyelination by morphological changes as you would expect it from the in vivo setting. And we could even show that myelin fragments, due to the demyelination process, could be detected inside of the microglia, which is obviously due to an active phagocytic state. So we've moved on, and we found out that, not really by chance, but luckily we could follow along the same lines, and we could grow cyno-iPS cells into a similar model, which also developed neurons and astrocytes over time. And again, the maturation markers nicely increased over the time. But also oligodendrocytes and myelin was present in this model. So this is now very promising for us. We are currently looking if we could also integrate cyno-derived microglial cells and make this a true human and cynomolgus two brain models for further assessment of our compounds. So in the last few minutes or so, I would like to give you an outlook and invite you for an upcoming opportunity in the area of teratogenicity, which is debatably maybe not only CNS related, but could be interesting. So, as you may know, teratogenicity are related birth defects, and mostly in the first trimester, the embryo is quite sensitive for these effects. Luckily, only 3% of the congenital disorders are due to compounds. But still, there is a strong need to assess compounds if they induce teratogenicity in women of childbearing age and pregnant women. So nowadays, we do this by applying animal studies, so-called EFD studies. However, they require the use of large number of animals, are very time-consuming, and that's why we only test in a very late stage of drug development the compounds usually, which is only during clinical trials mostly. And the human-specific effects, such as the thalidomide case, which is maybe the most prominent one, are hardly or sometimes hardly detectable in these species. So this, of course, is an opportunity for alternative in vitro models. Because, I mean, we could facilitate human-specific prediction, we can reduce the number of animals, and we can allow a decent throughput of these assays, enabling earlier testing, maybe even screening of compounds. But this comes along also with a challenge, because the data, as in the previous example, is mostly coming from animals, from the EFD studies, and we have luckily only very few examples from the humans. So therefore there exists something like I call it translational gap from animal EFD data to human in vitro models, because mostly the models we are having today are human-based models. So that's why some sponsors from bigger European pharma industry, together with NC3R, we developed a Crackit challenge, a funding competition in the area of in vitro developmental toxicity. So there is up to 1.1 million funding, which is unfortunately a little less than Rihanna presented, so there is no competition, of course. The launch event is on the September 6th And to get further details about the event and so on, please either register for the newsletter, or if you don't want to register, please have a regular look to the NC3R Crackit site. If you have further questions, you can also approach me and other sponsors during the meeting. So finally, the most important slides, obviously that work hasn't been done by me. Mostly it has been done by the people in the lab, which is, to name, I already called out Karen, but also Atina, Fulya have contributed a lot to all these models, and Christine, and the other colleagues from pharmaceutical sciences, the ophthalmology DTA, clinical safety, and also Manuela did a lot of work in the past years on teratogenicity prediction. And finally, I would like to thank you for your attention, and I'm happy to take questions.
Thank you very much, Stefan. It's amazing what you are doing in this field, yeah, to come up with these new models. Are there questions to Stefan? We have time for several questions.
I'm going to stand there. Hello, sorry, I was hidden. I have a practical question. How do you see the added benefit of having the Sino-MPS brain model on top of a human model?
Of the human model?
Yeah.
I think that the problematic is usually we assume that everything we see in the Sino, I mean, as we do for other species for safety testing, would one-to-one translate into the human. But if you come into very specific mechanisms of toxicity, sometimes there is a bit of a doubt. And that's why we really thought, because if you don't see, I mean, let's assume you have your human model, you start testing the compound which had some findings in the cyno, and you don't see it in your human model. Then is it due to the model, which does not recapitulate the cyno, or is it truly human negative, which means you could develop it further and bring it into the clinic? And this was an upcoming discussion every now and then, and that's why we really decided, as we could do it, why not creating such a cyno model? And that's where we are. And I think this really helps to better understand. I think the future is clearly having human models. But I think to me, there is a bit of a transition phase, right? In the meanwhile, until we get there, first we need to back-translate clinical data into our models to prove that they are truly relevant. But then also really show that these models, the systems per se, are relevant. And I know that the cyno model is not the same like the human model, but still it would give us further evidence that this kind of a human brain organoid model based on this protocol is relevant, and we can recapitulate findings which we may not can in the human.
Can I ask a follow-up question?
Sure.
Okay. And you're talking about back translation of clinical findings to your Cynos or to your MPS models. Have you tested a set of compounds which were kind of reference compounds to compare the performance of your human MPS in comparison with your Cyno and with clinical outcomes?
This is something we want to do, yeah. It hasn't been done yet. It was until now we really were in depth into the model development, but this is surely something we would like to do, yeah.
Thank you.
Further questions?
Really nice talk. This might be hard to answer, but is there any idea on how maybe these CNS organoids can model behavioral effects? Because I just think about moving away from animal altogether. Obviously, this is very maybe in the distant future, but you know, maybe earlier in the AOP, identifying changes, I don't know, neuronal firing or whatever. Has there been any thought about that? Because right now we're just strictly talking about killing mechanisms, including with the biologics, and there's a lot of biologics that don't have killing mechanisms. So I just wanted to hear your thoughts on that, the future of that.
Yeah, it's a good question. I think there are already models like seizure. I mean, at least there is some evidence that you can recapitulate at least some seizurogenic compounds. You can detect them in some more physiological, in physiological firing, electrophysiological readouts, right? But this is certainly something we need to look into. So most of the effects we are currently, at least for us at Roche, we are currently fearing is clearly neurotoxicity, which is direct or indirect toxicity of neurons and maybe related cells like astrocytes. And there are also some other things like biomarkers. I heard today a presentation about NFL, which is a prominent biomarker. I mean, this comes into play, and I think we also need to consider establishing those markers in our in vitro models and see, because there is a bunch of clinical data already around how well this translates. So this also brings maybe a bit more the physiological readouts together from a clinical or preclinical perspective towards the in vitro side, not only measuring ATP decrease or LDH release.
Thank you. Still touching upon the question that our colleague just asked, I mean, I think that it's really the elephant in the room. I mean, I think that we're making strides into learning how to understand biochemical toxicity. I mean, we have either structural or molecular readouts that allow us to tease that out. But when we're talking about the development of a brain, the subtleties of connectivity of cells and how that determines as to how the brain functions, I'm honestly not seeing yet the light in the end of the tunnel as to how we may be able to predict these subtle changes. And thus, how is it that we will be able in the foreseeable future to get by without having to resort to some sort of behavior testing in animal models? So it's a major challenge.
No, I think you're right. I mean, a lot of the neurotoxic deficits we see due to then behavioral deficits, what you said. And this is certainly hard, at the moment at least, in my opinion, to detect in the in vitro models. That's why I guess at the moment we try to capture at least what you said. I mean, the chemical or the mechanisms of several ideas of cytotoxicity being direct, indirect, immune-mediated or not. And the next step to me is, as said, leveraging a couple of the biomarkers which we can measure in the clinical setting, like NFL, YKL-40, and you name them, in these models. So at least, and then we build it from there. I think the field evolves so fast.
In full agreement, and I mean, and perhaps the key to this discussion is that all too often we think about the 3 R's and we make 2 of them irrelevant and we just think about the replace. I mean, what you're speaking to is a very powerful tool to very substantially reduce the number of actual behavior testing that would have to be done in animals, because I mean, if it fails by a biochemical toxicity screening, then I mean, you certainly do not, in vitro, you certainly do not need to pursue. So I think that's where the key is. I mean, the long-term solution, yeah, it's still out there.
I agree, and I think we should not say that this is a solution which, you know, replaces everything. So it depends heavily on the context of use and of the question you're asking. And I would never claim that this is a full brain model, right? But rather this addresses currently the questions we had with regard to the portfolio aspect we were working on. But this is certainly something, yeah, we need to work on it, get there.
Really good work. Thank you.
Thanks.
Okay, so one last question maybe.
Hi, that was an excellent talk. I was curious about your thoughts, some of the new literature coming out with aspects of neurodevelopmental disorders, even neurodegenerative disorders, where the etiology of those disorders are much earlier in development, or at least in some of these early neurodevelopment models we can see phenotypes with iPSC lines. What are your thoughts around sort of using maybe those models that are arrived at faster or derived faster to do some sort of preliminary screening of therapeutics, and then maybe using that to validate in the much longer, more costly developed organoid models?
So if I got it right, your question is around neurodevelopmental toxicity testing. I think it's clearly all the models, the IPS derives, they seem to be still kind of immature, right? Or at least having a degree of immaturity. But I mean, in our context, we always want to have real mature models. So this is rather a bit something we are struggling with, because all our questions, at least for us, we are currently addressing, we want to have the most mature brain, retina, and so on, neuro tissue we could achieve. So this is certainly of interest, but at least for the last 5 to 10 years I was working in this field, I mean, we haven't come across the neurodevelopmental toxicity aspect.
Okay, so thank you. Thanks again, Stefan.